Hi, this is Miss Slitton, and this is my wonderful Period 1 APBio class. Say hi. And those of you who are starting your Thanksgiving vacation a little early, anyway, we miss you and we're jealous. We're starting in on Chapter 16, How Populations Evolve. The very first thing we're going to do is start by watching this video, The 5 Fingers of Evolution. I'm going to pause this. I recommend, if you're in my class or not even in my class, that you watch this video. It's a really good little TED Talk. And so I'm pausing. And wait, before I pause, take a look at the lab. We're going to start the lab on Monday when we come back from Thanksgiving break, and it is a team lab. So you can talk to your buddies in class to make sure you're ready to go for that, okay? All right, I'm going to little pause. Pause. Okay, welcome back. I hope you watched that video. So first thing we want to talk about is when we look at antibiotic resistance of bacteria. This should be concerning to you. We're doing different things. We're selecting for bacteria to be resistant to antibiotics by putting so many antibiotics in our environment, yes? Evolution. Could I please have oldest bio buddy give the definition of evolution as it is up here? Go. Okay, so you could have one person who's very, very tall, okay? We talked about this before. But that doesn't mean evolution has occurred. That's just one mutant, potentially, right, who's super, super tall. Okay? When you talk about evolution, you need to look at it as a population evolving. And so when you look at a population, who is it? Old, go. Yeah? Young, go. What's the population? All right. So on your notes so far, a temporary or seasonal change during an individual's lifetime is not evolution. It must be heritable, which means you can inherit it, right? Heritable. So evolution is a change in traits governed by alleles. What is an allele? It's what? A variation, right? Remember we said, like, if I had big T, little t, okay, that's a variation on a gene that we are looking at, right? That's an allele. Remember when we talked about multiple alleles? What was an example of multiple alleles? I can't hear it. What? Oh, that's polygenic. Yes. Blood type. Why is blood type a good example of multiple alleles? Because there's big I A, big I B, and little I. That's multiple alleles, right? Polygenic is when you have like I's, Q's, R's, P's, L's all contributing to a trait. So what evolution is a change in the allele frequency. If the allele frequency changes, evolution is occurring. Yes?
Is hair like the wavy and straight? Is that multiple allele?
No. Does anybody remember what wavy and straight was? Yeah. Intermediate. And that was when the hybrid has its own phenotype. Okay. Incomplete dominance. All right. So a change in traits governed by alleles in a population, a group of a single species living together in the same geographic area as a population, specifically the change in allele frequencies, a change in the allele frequencies in a population over time. Specifically, the change in allele frequencies in a population over time. What we just watched in the video are 5 different items that can change or alter allele frequencies. Not really non-random mating. We'll talk about that. That could change genotypes, which over the long haul could change allele frequencies, but we'll talk about that in a minute. Okay, specifically the change in allele frequencies in a population over time. So there's 2 kinds of evolution we want to differentiate between, and the dark-shirted bio buddy would like to differentiate between them. Go ahead, dark-shirted bio buddy. Go ahead.
Okay, so macroevolution are the processes through which new species arise.
Okay, so microevolution is within a population, Macroevolution is large scale, forming a new species. So let's look at some microevolution that could very well lend itself to macroevolution. This is a yellow-bellied three-toed skink. And in higher elevations where it's colder, some of these yellow-bellied three-toed skink are starting to give live birth. So they're holding their, developing, they're not laying eggs, they're holding them in until it's time to give birth. Why would that be advantageous adaptation? Yes?
The cold might kill the egg.
Cold might kill the egg. Yeah, it's more protected. Wherever they go, the eggs are with them. So right now, this is microevolution because they will still mate with those that don't carry their embryos inside. But if over time, the embryo-carrying ones only mate with other embryo-carrying ones, then we are isolating it due to that specific trait. So then that would not be microevolution, that would be macroevolution. That would be macroevolution. All right? So what we're studying in this chapter is population genetics. It's where you look at a Punnett square, not between two individuals, but basically the population Punnett square. Okay? Everybody interbreeding with one another. When you talk about that, we're talking about the Hardy-Weinberg equilibrium. And Hardy-Weinberg equilibrium has some assumptions you have to keep in order to maintain it. But for right now, we're just going to learn a little bit about it. So we're looking at the alleles that govern the traits. So let's say we have these wild boars. And we can see their genotypes, yes? Okay, so each individual is diploid. They're carrying two of those of each copy, right? Because they're diploid. So let's see, if they took both of their copies and threw them in a pool, that's what we would call the gene pool. And so in the gene pool, for that entire population, we want to know what is the frequency of each allele. So if we take all our alleles, throw them in a pool, what's the frequency of each allele? So let's count up how many big B alleles do we have and how many little b alleles do we have. Do that with your bio buddy right now. Okay, so how many boars are there? 7 boars, right? So if each of them carries 2 copies of the allele, then how many alleles do we have total in our gene pool? 14, right? We each take both of our alleles, throw them in the pool. Throw them in the pool. Okay, so we have 14 total alleles. How many of those 14 are big B? 7. Okay, you counted that because you said 1, 2, 3, 4, 5, 6, 7. Now let me ask you this. If you know there are 14 total alleles, then you know if 7 of them are the big, then it has to be what? 7. Okay, and that's one of your Hardy-Weinberg equations, is this first one which says P plus Q equals 1. Okay, well, those aren't any B's in there, but it says P plus Q. You know what P stands for? The dominant trait. And Q stands for the recessive trait. So all together, those are my total amount of alleles. I've got 14 alleles to deal with. So if I've got 7 of one, I've got 7 of the other. If I had 10 big B alleles, I would have to have 4, right, of the little. So I have, this tells me right here, of this, what is 7 divided by 14? 1. Okay, and we'll say 0.5, and then again 0.5, right? So of the total alleles we have right here, 1 equals P plus Q, right? 1 equals 0.5 plus 0.5. Are we all right with that? That is what's called the allele frequency. We just calculated it out. Okay, the frequency of the P allele, the dominant allele, is 0.5. Then the frequency of the recessive allele is 0.5. If the frequency of the dominant allele is 0.7, then the frequency of the recessive allele would have to be what? 0.3. So when you do Punnett squares now, okay, we don't do Punnett squares with individuals, we do it with a population. Okay, so now basically you are crossing the allele frequencies of each of those alleles. So we said P was 0.5, then Q is 0.5, and Q is 0.5. Okay, this is how you get to the second equation that we're working with. What's P times P? 0.25, and P times P is P squared, yes? That's like being big B, right? The two dominant ones. Okay, so this one is 0.5 times 0.5 is also 0.25, but this would be what? P times, what would that be? P times Q. This would be like our heterozygotes, yes? Okay, so again we have 0.25, because we're crossing the whole population with itself, and then we have another PQ, and what's this last one going to be? Q squared, and what's this number going to be? 0.25. If I write this as an equation, now I'm not looking at the allele frequencies, I am looking at the genotypes. Okay, so once I have my gene pool, what this means is once you know what's in your pool, you can predict what genotypes you will get out of that. Or if I know what genotypes I have, I can predict what's going to be in my pool. Right? I can go back and forth. So this equation right here is P squared plus how many PQs do I have? 2 PQs plus Q squared equals 1. Now, decide with your bio buddy which parts of that equation would display a dominant phenotype and which part of this equation would express a recessive phenotype. Discuss with bio buddy. So which ones would show the dominant? P squared. Who else? 2PQ. And these right here, they would show the what? Recessive. So when you look at a trait, you cannot predict. You cannot sit there and go, oh, if I look at the dominant trait, I don't know. They could be homozygous dominant or they could be what? Heterozygous. Okay? The only one I'm going to know for sure, right, and we've talked about this before, right? Remember the whole need for test crosses? Because if somebody's brown eyes, we don't know if they're brown-brown or brown-blue. The only ones we know for sure are the blue-eyed, right? Now, I don't know all your genotypes. I don't know, but we could predict what everybody's genotype is, right, using these two equations. So how many people are in this room right now? 2, 4, 6, 8, 10. 2, 4, 6. Oh, wait. 2, 4, 6, 8, 10. 2, 4, 6, 8, 30. Okay, is that what you said? 30. Perfect. Okay, now, of this population, okay, you're either gonna be brown eyes, you're gonna be blue eyes. If you're green, we're gonna throw you in with blue. Okay? If you're green, we're throwing you in with blue. Okay, so of our 30 people, okay, we have 30 people in this room. So that means how many alleles are we carrying? 60. Okay? So, but we have 30 individuals. Let's find out what the percentage is of our 30 people. I just wanted to remind us we have 60 alleles out there. So if you have blue eyes and you're a student in this room, please stand up. If you have blue eyes. Okay? So what percentage of our population, wait, keep standing up. How many blue-eyed people do we have? 1, 2, 3, 4, 5, 6. Do you agree with that? So 6 of our 30 people have blue eyes. 6 of our 30. So how many people have brown eyes? 24. Now, those 24, what could their genotype be? So they could be P squared plus all of our 2 PQs, right? And the blue eyes, though, what's the only thing they can be? That's all they can be. Because in order to have blue eyes, it's recessive, yes? Okay, go get a calculator. Either pull out your phone or go get a calculator in the back, and let's figure this out. Let's figure this class out right now. All right. So now I know P squared plus 2PQ plus Q squared equals 1, right? I know that. This is just saying homozygous dominants, heterozygotes, homozygous recessives. I want to solve for the allele frequencies in the room. So first of all, I need to know 6 divided by 30, what percentage is that? What is it?
20.
Okay, now take the square root, right? That will help me if I know q squared, right? Q squared equals what? 20, right? 0.2, was it exact?
Yeah.
Okay, so now take the square root. What is q? 0.45?
Yeah, 0.45.
0.45. Okay, so if I know q is 0.45, right? We know that all our alleles have to add up to 1, right, in our gene pool. So if q is 0.45, what does p have to be? Point what? 0.55. Now, that's what we are predicting based on the number of blue-eyed people in this room. That may not be what we're calculating now. These are just predictions based on the amount of blue eyes because we know those are the only two options here. So now I can solve for how many of you are probably homozygous brown and how many of you are heterozygous. So to find out how many of you are homozygous brown, what would I have to do? Take 0.55 and do what to it? Square it. So give me a percentage. Is it 30? Okay, so 0.3, so now I could actually see how many people in the room, right? Because now I need 30% of how many people do we have in this room? 30% times 30 people in the room is going to be what? 9. So I am predicting of the 24 of you left in the room that have brown eyes, I'm predicting 9 of you are homozygous brown. Ah, where are you? Okay, now 2PQ, right? 2PQ, 2 times 0.55 times 0.45. That'll tell me I'm predicting how many of you are heterozygotes. So do the math on that. 14. Now we could figure it out, right? We have to have whole numbers for people by how many we've already said that are homozygous recessive and how many are homozygous dominant, right? Okay, so Hardy-Weinberg says it just gives you a way to mathematically model. Here's the biggest point of it, though. If the frequency of these two alleles, 0.55 and 0.45, if, let's say, Q increases, let's say Q increases to 0.6, that means P has to go down to what? If Q increases to 0.6, P would have to go down to what? 0.4. Did the allele frequency change when we do that? Yes. Here's evolution is the change in allele frequencies. Evolution would have occurred. If we're still the same species as a result of that, that change in allele frequency would result in what kind of evolution? Microevolution. But if blue-eyed people start only having sex with blue-eyed people, and brown-eyed people start having sex with brown-eyed people, it could lead to speciation, which would be a form of what? Macroevolution. Okay? Wrap your brain around that and talk about it with your bio buddy. Put it in your own words. Say, whoever feels the weakest, you're the one who should talk. And then the stronger person, like, hey, I feel weak, you know. But if you feel strong, both of you talk about it. I don't care. But try to help them out. And I'm going to give you these two equations to help yourself. Go ahead, talk about it. Use these two equations to talk about it. All right, now let's go to our notes. Okay, microevolution in the peppered moth. We're going to get to that in just a minute. Population genetics, studies of the diversity of populations at the level of the gene, how it changes. We look at allele frequencies. The gene pool is all the alleles in a population. All the alleles in a population. Blue take over B and slate take over C. Go. So genotypes are as a result of the allele frequencies. Genotypes as a result of the allele frequencies. So if you're dominant, like little p, another way to write that is p squared, then that's homozygous dominant. If you're PQ, you would be identified as a heterozygote. If you're Q squared, you're a homozygous recessive. If the allele frequencies for a particular trait remain unchanged, P and Q values remain the same generation after generation, then that trait is not changing or evolving, and it is said to be in Hardy-Weinberg equilibrium. All right, let's do some practice. I went and got some pea plants. The yellow flowers are dominant over the pink flowers. Let's see if we can calculate the allele frequencies. So I want to figure out the allele frequencies of P and Q based on this picture right here. And I'm going to pause this for a minute while you do it. Okay, welcome back. Hopefully you took a minute to figure that out. So I told you in this case that pink flowers were recessive, right? And that's key. Because you know these yellow whitish flowers could be what? What would be the yellow and the whitish flowers? They could be homozygous, right? P squared, homozygous dominant, or what? Or they could be the heterozygote part of the equation, yeah? So the only one I could solve for sure, because I don't know about the rest, the only one I know for sure would be the Q squared, right? That's the only one I know for sure, because who represents Q squared up there? Pink. So I have 1, 2, 3, 4 pink flowers. Now how many plants? How many plants do I have total? 12. So I know my Q squareds are 4 of my 12. So I do the math for that. What is 4 divided by 12? 0.33. So I know 33, all that's telling me is 33% of them are showing the recessive trait. That doesn't give me Q, that just says 33%. So in order to solve for Q, what do I need to do? I need to take the square root. So when I take the square root of Q to get the allele frequency, what does that equal? 0.58. Okay? So if I know Q is 0.58, that's the frequency of that allele, I know a second equation is P plus Q equals 1, which means if, equals 1. So if I know Q is 0.58, then P has to be what? 0.42. That's how I solve for P and Q. Now, I could take it a step. First of all, just check with your bio buddy that you understand how I went about that. I don't know who these guys, I don't know their genotype. I'm just predicting, I know they're the dominant trait, but they could be homozygous dominant or heterozygote. Who can I cross them with to find out what they are? The pea flower, right? But I don't know. So I'm just predicting what I think it is. So if I wanted to figure out, if I wanted to predict, I don't know. If I wanted to predict how many of them are homozygous dominant, what would I need to do? If I wanted to predict how many of them are homozygous dominant. Okay, so the homozygous dominant ones are going to be what? P squareds, right? So I would take this value right here, 0.42, and square it, right? That would give me how many of them are P squared, a percentage. So take your 0.42 and square it. What did you get? 17.6, right? So now if I wanted I know 17, 18% of them should be homozygous dominant. So how many pea plants up here? What would I do? I'd take that number times how many plants do I have? 12. So I would predict maybe about 2 of them are homozygous dominant. That's what I'm predicting. Do I know for sure? No, I don't know for sure. But I'm predicting 2 of these are going to be are going to be homozygous dominant. Then the rest of these must be what? PQs. Right? That's got to be a PQ, that's got to be a PQ, this is a PQ, this is a PQ, because I know these are Q squareds, right? The pink, these. I know they are because that's the only way they can express that trait. Okay, check with BioBuddy, see how they feel. All right, questions you want to ask me? All right, let's look at this. So allele frequencies, population genetics, once again, studies the changes in the genetic makeup of a population. The gene pool is all the alleles of that particular population. If it changes, in this case, we had an allele frequency of 0.6 and 0.4. You all right with that? Okay. How many of them would be heterozygotes? How many of this population would be heterozygotes? How many? Okay, what's this one right here? This is homozygous dominant. This would be homozygous recessive. So how many of them are going to be heterozygotes? 48%. So if I had 1,000 individuals, how many would you predict to be heterozygotes? 480, right? If I had 1,000 individuals. Okay, so if big L became 0.7, then if that became 0.7, and then this would have to be what? 0.3, right? And this is 0.7 and this is 0.3. This would be then what? 49%. This would be 21, right? 21, and this would be 9%, right? Then I would say how many of them? 42% should be heterozygotes. If the allele frequency changes, what is occurring? Evolution. All right, perfect. So let's do a little bit of practice here.
What are we today?
So let me take this out. What did I say we were going to be today? We talked about it in the review.
Sandwiches. Yes, sandwiches.
Favorite sandwiches? Really? I don't even know what you meant.
Like how specific?
Okay, but your favorite sandwich. Oh, now I remember. Didn't we? Favorite sandwiches.
Oh, I remember.
Yes. That's what I was saying, yeah.
I like those things. Yes. Oh, there it is. There we go. A burger is
There we go. Winnie Witch. I knew it was coming.
Oh no.
Boneless, of course. What is it? A Kinzo? Jane Chen sandwich. All right, let's go. A couple more of you. Hurry up. Okay, now I encourage you to do these independently. Okay, and then check with your BioBuddy if you want to, but I just, try it independently. And I'm going to put a pause here. Actually, I will show the questions for those of you at home. Oh. Oh. Okay, I am going to pause. Okay, we are going to go ahead and stop and work through these in. Okay, 4% of the members of a population of pea plants are short. Recessive characteristic. What is the frequency of the recessive allele? Yeah. Not how many? This is what most people put. That was 2, and that was 2. 4.
Oh no, the 20. We got %.
Okay. Yeah, these people, they could get it.
Yeah.
2. I give that to you. Okay. So let's talk about how we solved that problem. 4% of the members of a population of pea plants are short. What equation are we talking about? If I'm looking at pea plants, I'm looking at a whole plant. So am I looking at allele frequency or am I looking at a genotype? I'm looking at genotype. So I'm looking then at this equation right here, right? These are genotypes. Homozygous recessive, heterozygous, homozygous dominant. So it says 4% of the members of a population of pea plants are short. So that's a recessive characteristic. So which part of that equation am I looking at? Q squared. So Q squared equals 4%, right? So how do I write 4%? 0.04. If you're not sure, you could, I don't know. Okay, then what you want to do is you want to take what to solve for Q? I want to take the square root of that, and when I do that, Q equals what? Yeah. Try it on your calculator. Just double check, okay? Make sure that works for you. All right, let's look at the next question. Terrifying. 4% of the members of the population of pea plants are short, recessive characteristics. That has not changed. What is the frequency of the dominant allele? Well, if the recessive allele, this makes sense, right, because of the amount of people that missed it before. If Q is 0.2, then P has to be what? 0.8. And you guys right here, you two, you get it right too. Okay? Discuss with your bio buddy right now. Where did you go right? Where did you go wrong? Just on those first two parts. Go ahead. Let's talk about it. Oh, all right. Then I asked, same thing. 4% of population pea plants are short, recessive characteristic. Didn't change anything. What is the predicted percentage of individuals that would be heterozygote? That would be 2 times P times Q. So, so that would be 0.2 times 0.8 is 0.16 times 2 would be 0.32. Okay? Now, that one would come and bite you in the ass, right, if you got the first one wrong. Because if you started off getting the first one wrong, then you got the next two problems wrong, right? So we got to be careful on our math. That's what that should remind you. Be careful on your math. Now, okay, I have no idea. Look to me just for a minute. You are going to be tempted. I have given you resources of more practice problems for you to do. So you can practice this some more. I promise you that is coming your way. It's already in there. It's embedded in. It's baked in. But right now, what we want to do is say, yes?
For number 2, wouldn't you just do 0.96 squared? Or 0.96?
I can't do that because I can't take the square root of that because the dominant trait represents both the homozygous dominance and the heterozygotes of which Q is a part of, right? That's why you must always use the blue-eyed people, right? You have to use the recessive trait. Think of it in something that you can relate to. Because we don't know if the brown-eyed people in here are brown-brown or brown-blue. So we can't take the square root of them. Okay, because it would be a square root of P squared plus 2PQ. It's just easier to take the square root of the recessive trait, right? Okay. Now, what we want to do is, what do we do with this? So you talk about Hardy-Weinberg equilibrium. It's saying if all the conditions stay the same, okay, then I can predict these allele frequencies, and if the allele frequencies stay the same, then evolution is not occurring. Well, that, if you can freeze the allele frequencies, that used to be our governor, if you can freeze the allele frequencies, you can stop evolution. If the allele frequencies change at all, then evolution is going to occur. And let me tell you, it's very difficult to freeze allele frequencies. Remember the 5 fingers he did? Those 5 fingers of items can alter the allele frequencies, and therefore evolution occurs. That's the point that gets made by this. So, for instance, the very first condition you have to meet is no mutations. Okay? But mutations happen. Okay? So, if you're looking at your notes, I gave you the Hardy-Weinberg equilibrium. And it says the Hardy-Weinberg principle only applies if the population remains unchanging. To ensure that 5 conditions must be met. No mutations. When he did the finger one, what was the mutation? What finger was that? Middle. Okay? The middle finger mutation. So that'll help you remember that. So no mutations. The second condition was that there was no migration or gene flow. which one of the fingers was the no migration or no gene flow? Yeah, because it's pointing. You go over there, you go over there. No gene flow. Okay? And we're going to kind of flesh these out a little bit more, but this makes sense to us, right? You know mutations. If you spontaneously mutate from brown eyes to blue eyes, and by the way, blue eyes is a mutation, right? Because we have lack of pigment here to protect us from the sun's rays. So all of us, 6 plus me, we're all blue eyes. We're mutants, okay? Because what happens is light goes in and it can damage our retina. So that changed allele frequencies because now that's part of what's in our population. If you have gene flow, if somebody comes in from another country, if we have a huge influx of people coming from another country and we're interbreeding here in Oak Park, could that change our allele frequency? Yes. Okay? And if we had people leaving, exiting, that could affect it. So gene flow definitely changes that. You know what those are right there? See the stingrays? And then the next one is no genetic drift. Genetic drift has to do with chance affecting it. And so in order to avoid chance having a big influence on your population, if your population is large, you avoid chance influencing your population. So on your notes, large gene pool. So no mutation, no migration, large gene pool. And I'm going to talk more about chance here in just a minute. This is just an overview. The fourth condition to freeze allele frequencies is random reproduction. It's funny because they're bugs. Okay, so this means no bug is greater than another. You'd be like, any old bug can come have sex. Okay, so this is full-on random reproduction. Okay, please don't be random in your reproduction. Please choose wisely. Okay, but if you start selecting for a particular trait, that could eventually lead to speciation if you isolate yourself and only, like I said, tall people have sex with tall people, short people are blue eyes with blue eyes. And the next one is no selection. Why did I pick this picture? What is this? Cobra. What's on the back of his top of his head there? What's on the back? What does it look like? Eyes. Do you think he's ever seen those eyes? No, he's never seen those eyes. Why would you see that adaptation in the cobra? Why do you think? Okay, so if a predator comes up and a predator is going to try to eat this snake, do you think he's going to like skip up to the front of the snake, say, hey, hey, snake? No, because what will the snake do? Bite him. So the trick would be to come up, what? From the back. But if he sees those eyes, maybe he thinks the other side is the back. Do you see? So over time, those cobras that look like they had eye spots on the back of them, those are the ones that survived and reproduced. And it got selected more and more and more and more because somebody would come up on this side thinking, those are the eyes, look, it even has a nose, and then, okay, and they get bit. All right? So that would be an example of selection. And selection could change the allele frequency over time, so that's why it's no to selection. So on 5, no selection. So it only matters that the allele frequency remains unchanged. The genotypes can change due to non-random mating, but that might change here and there. And I'll explain that in just a little bit more. But you want to keep the allele frequencies the same, and these are the things you need to do in order to do that. Not it. Ping pong back and forth. These are the 5 conditions you have to meet. Go ahead. All right. So, it is hard to meet all 5 of these conditions. So since it's hard to meet all 5 of these conditions, therefore evolution is occurring. Because it's hard to meet all 5 of those conditions. So that's your E. Since it is difficult not to violate these conditions in natural populations, alleles must be changing and therefore evolution must be occurring. I like this picture. All right. So what type of evolution is this? Coming back to the title of what we said in here about the peppered moth. I mentioned this in a chapter before. This is industrial melanism. Do you see the moths on the tree?
Yes.
Okay. There's a light colored moth right here. Do you see him? And here's a dark colored moth. Now, who do you think the bird's going to see? The bird's going to see the dark colored moths. So what do you think is going to happen to the allele frequency of the dark colored moth? It's going to go down. Now, post-industrial revolution, there is soot on the trees. Now who are you going to see? The white. So this could be a change in allele frequencies, but it's not changing them into two different species. So what kind of evolution would this be? Yes, this would be microevolution. Okay, because it's just changes within your allele frequency due to the conditions or the environment. But if it eventually leads to only the dark moths have sex with the dark moths and only white moths have sex with the white moths and they accrue some other changes and mutations and other selection pressures, that can lead to what kind of evolution? Macroevolution. Good. All right. So now, which of these can change allele frequencies and therefore cause evolution? We talked about the first one, mutations. So I'm just fleshing a few of these out. There's a few more details on them. So go to your microevolution and Hardy-Weinberg equilibrium. By the way, your next chapter that we get into next is going to be about speciation, formation of new species. That's what's coming up next. The opposite of the 5 conditions above must therefore cause alleles to change and therefore evolution, mutation. Defined, a change to the DNA sequence can serve as a source for variation. Blue do B, slate do C. Now, take a look right here. This is something we have already studied, right? We studied this a chapter or so ago. So here's the original DNA template. And then notice the 3 bases, right? They code for the amino acids. And in this case, instead of being TACT, it was TAC what? A, which then coded for U. So you got a different amino acid here, serine. And in this case, what does this look like? Must have been a frame shift. Okay? So mutations can change allele frequencies because they change the proteins they code for. Okay? Second one. So that could definitely lead to evolution. The second one is migration or gene flow. Any new genes in or any new genes out. So that's migration. Gene flow is movement of alleles between populations. So mutations and then gene flow. What's the third one? What's this one about? What was the pinky about? Yes. Small populations can lead to genetic drift because chance can have a higher impact on small populations. If you have a population of 20 tigers, okay, and they are living in a volcanic, what's it? Crater? Not crater, what do you call that? Maybe let's go basin. Okay, and you only have 20. And then there is some kind of explosion or something that happens, some earthquake opens up a little fissure, lava comes out, or it has another volcano comes out again. You can lose those 20 like that, right? It's done. And so genetic drift has a bigger impact on these smaller populations. Now there's 2 types that you talk about. One is called the bottleneck effect. Now, bottleneck effect, we would think about this really about the freeway. They'll talk about bottleneck effects, right? So if several of the lanes are closed off, only a few can get through. So if you look at this jar right here, there's all different colors, but because of the bottleneck, only a few get through, and that may not represent the total population. So in this case, you have brown frogs and you have green frogs. And if a tree happens to fall on the brown frogs and all the brown frogs die, then all you have left are the green frogs. What? What? Okay. So this is a form of genetic drift. So look at your notes. Genetic drift Changes in allele frequencies of a gene pool due to chance events. Smaller gene pools are more impacted by drift. A bottleneck effect is when there's a disaster. So new population's allele frequency does not reflect the original. And so that's the bottleneck effect. There's another one called the founder's effect. It is another form of genetic drift. Now, Amish people migrating to the United States, it just so happened that they had a six-fingered dwarf in their midst. And so when they migrated to the United States, Amish people tend to breed just with other Amish people, right? So there is a higher incidence of six-fingered dwarfs in Amish. Can you see the six fingers? Yeah. Okay, of six-fingered dwarfs, and this actually, both of these kind of travel together, this trait. And so because of that, there's a higher incidence of six-fingered dwarfism, because that is the population that founded this new area, and they just bred with themselves. Take a look at this room. Okay, do we represent the United States here? Do you think this is the general population of the United States? This probably not, right? So if everybody in the United States died except for us, okay, and we were the Adams and Eves of a whole new generation, would that accurately reflect the original population? No. Okay? All right, so this is a form of genetic drift. So founder's effect, come back to me. Portion of the population starts a new population with a fraction of the total alleles. The new population's allele frequency does not reflect the original. So which form of genetic drift is this? He says, why doesn't anybody love me? Looks like we're the last two. Oh, great.
Oh, great. Oh.
So what is that? What do you think? Bottleneck? Yeah. Yeah, I would say it's because they went through a crisis and there's a few left, and nobody was normally mating with this one, but now, okay, that's all I have left, so okay, I'll mate with you. All right, and then the next two kind of overlap, but it's about inbreeding, and I wouldn't recommend it. But inbreeding is going to have a higher impact on smaller populations. It doesn't necessarily change the allele frequency. It just combines them in a particular way or a particular fashion, but it doesn't. It's not going to. It could change the genotype, but not actually change the allele frequency. So inbreeding, mating with relatives, doesn't affect the allele frequency, but can ultimately affect the genotypes. And then phenotypes, which can be selected for or against. You could select against them. Like if due to your inbreeding, you ended up having sickle cell, or you had hemophilia or something, not sickle cell, but well, yeah, you could get that too. You had hemophilia, right, due to inbreeding, and then nobody mated with you because you were weaker, because you had hemophilia, then you could eventually then have a change in allele frequency. Would you ever be able to completely wipe out a recessive trait? What do you think? Could you ever completely wipe out a recessive trait?
Yeah.
You can make it small, but where can it always hide? In the heterozygote. But you don't know. You don't know. Okay, two people. Okay, that scenario. All right. Now, it's funny because they're lions. This may not change. Random mating is, okay, whoever, I'm game, okay? But this is non-random. This is purposeful mating. In your population, it doesn't necessarily change the allele frequencies. It's just within your population, some tend to mate with others, and they click, they're in a clique of who mates with who. But it doesn't mean it will necessarily change the allele frequency, so this may not lead to evolution. So on your non-random mating, I gave you A and I gave you B, so Slate does A and B does Blue. Go. your favorite?
The bird. No, the big one.
No, the other one.
Okay, it's called assortative mating, and it can be important for initial divergence and then maintenance of the species wanting to mate with somebody just like you. But unless they only mate with that one over time, it's not going to lead to any kind of speciation or change in allele frequencies. All right, and then this brings us to, non-random mating. Which finger was that? The ring finger, right? So this was small populations can lead to change, non-random mating, mutations, gene flow, and then this one is, the thumb is natural selection, okay? So natural selection is the one that leads to adaptation. Now, chance can have an effect on speciation, but probably natural selection would have the biggest impact on evolution. And we've talked about the process of evolution before and why you would be selected for to have that long, narrow beak over time. We talked about the way Lamarck and Darwin would talk about longer giraffes. Dead giraffe. Okay, so anybody with his back. Okay. Oh my gosh, watch out, elephants. Okay, 5A. Watch the news. You'll understand my rage. 5A. Some phenotypes have a reproductive advantage. Their traits will be passed on while others will not. Natural selection is the foundation of Darwin's theory of evolution. Okay, and then I'm going to stop this right here.